Infectious diseases have significantly contributed to human mortality throughout history. They were the leading cause of death until the discovery of antibiotics and remain a major concern today, as sadly reminded by the recent coronavirus pandemic. Treponema pallidum is a bacterium responsible for various diseases in humans, including syphilis, a sexually transmissible infection currently on the rise worldwide, and other treponemal diseases endemic to specific tropical regions. For a long time, researchers have debated the origin and evolution of this pathogen, proposing various hypotheses. Did the pathogen evolve alongside humans for a very long time? Or did it first emerge in the Americas and then spread globally in the wake of colonialist expeditions at the turn of the 16th century? For decades, researchers have concentrated on analyzing historical records or studying pathological alterations in bones from archeological sites, seeking characteristic changes caused by the microorganism's infection that could provide evidence to support one theory over another. Despite considerable effort, the current knowledge seems insufficient to settle this matter. A new perspective was needed to resolve this decades-long debate.
To unravel this enigma and shed light on the origin and evolution of this pathogen, an international team of researchers adopted a novel approach. Their objective was to extract the pathogen's DNA from ancient human bones.
Hello everyone, I am Bozzi. I am a researcher at the University of Lausanne in the Computational Biology department.
Together with our colleagues in Colombia and in the United States, we have tried to provide the first genomic evidence of Treponema pallidum from ancient South American populations.
Ancient bones can contain both the DNA from the human and the microorganism that coexisted or infected the individual. Today, thanks to new methods and technology, we can extract and sequence this ancient DNA. However, due to age and biochemical processes, this DNA is usually highly degraded. For this reason, we need specialized labs that minimize the risk of contamination with modern DNA. Each organism's DNA is unique, and by reading its sequence, we can identify the species to which it belongs. In this way, for example, we can identify pathogens that infected ancient individuals. And this is done by using specialized bioinformatic algorithms. Furthermore, the study of ancient DNA from microorganisms can allow us to trace their relationship with present-day pathogens. And this can be done, for example, using phylogenetic trees.
In our study, we extracted the DNA from a 5,000 years old individual from present-day Colombia.
This hunter-gatherer lived in rock shelter in what is now the savanna of Bogota. Surprisingly, we recovered enough DNA to reconstruct most of the genome of this ancient pathogen. The genome is the entire DNA sequence contained in a living cell, and it holds all the information to make each living thing what it is. By comparing the ancient Treponema pallidum genome with modern ones, we gain insight into their relationships. We observed that the ancient genome represents an ancestral form of Treponema pallidum that diverged long before the emergence of modern strains found today around the globe.
These results mark a significant step forward in our understanding of how this disease first emerged and diversified.
We now know that an ancestral form of Treponema disease existed in South America as far back as 5,000 years ago.
This pathogen was quite different from modern ones, having diverged long before the diversification of modern strains.
The study also emphasizes the revealing potential of ancient DNA in advancing our understanding of human health, diseases, and the evolution of microorganisms.